Our main interest is occupational asthma in this model, and we study that via a small chemical TDI, toluène diisocyanate. So occupational asthma, I just introduced it a little bit, but you all know that. But what is important here is the airflow limitation and airway hyperresponsiveness due to a case being present at work and not necessarily at home. And it's a very important disease, so 9 to 15 people having asthma have a kind of occupational asthma, and therefore it's one of the most common work-related diseases. So I'm not going to go in details here due to time, but it's important to know that asthma is a very complex, it's not one disease, it's more a complex of different inductions to more or less the same phenotype. And you can have an induction via large molecules, proteins, but you can also have an induction of asthma due to low molecular weight compounds, as for instance TDI, which the molecule is here. And you have different outcomes which can be IgE mediated or rather not clearly IgE mediated. So, and diisocyanates, as you all know, and I'm not going to go into details, is an important group of chemicals in industry to produce a lot of different compounds, coating materials, foams, and so on, but also in house it's been used in consumer products where you can have release of different types of isocyanates or diisocyanates. So, and what was now wondering us 10, 15 years ago was the observation that the development of chemical-induced asthma, which was already being quite strict in the diisocyanate industry to really reduce the exposure via inhalation. So because the immune response was via the respiratory tract, and the focus was really on the regulation, prevention of airborne exposure. But still, as you see here, this guy is wearing a mask to protect himself. But despite this, still a lot of people got asthma due to exposure to diisocyanates. And therefore, we started to focus also in a mouse model on skin exposure. And then you see the difference that this person is not wearing protection for his skin, while in this application, this person is clearly aware that maybe also induction due to skin exposure could lead to asthma. And therefore, we built this mouse model in which we expose the mouse on the skin of the ears, day 1, day 8, and that's a sensitization step. And then we do a challenge of the mouse, of the lung, at day 15. And then we can measure early responses at the day itself, directly after exposure, or we can measure late responses, airway hyperactivity, and look into different parameters after sacrificing the mice. And in this model, we have clearly an induction of asthma due to skin exposure. And then, after developing the system or the model, we wanted to look into, can we find any biomarkers which can maybe predict very early in the process of a person getting sensitized to these chemicals. And therefore, we did choose Proteomics, which is a global strategy in which all proteins, so the proteome, derived from cells or whatever tissue you'd like to take, to visualize that and to see changes due to exposure to the compound of interest. And therefore, we set up an experiment where we would like to look at changes during sensitization, not the asthmatic response, but during sensitization, whether or not we could pick out maybe some molecules of interest. And so I again show you our global scheme, so sensitization and challenge. And what we did do then is we sampled at day 4 or day 11. We sampled the lymph nodes draining the ears, so the auricular lymph nodes in which we expect, and it's clear that there are the most changes going on of the immune system in these lymph nodes draining the site of exposure. And we also looked into serum, and we did do a proteome analysis. So before putting everything in proteome analysis, we checked whether or not our mice did really get some sensitization, and you can see here. So first application control, because this stands for acetone olive oil. That's the vehicle we use to dissolve our TDI in, so that's the control. And so one application day 4 or day 11, and we see clearly an increase in weight of the lymph nodes of interest. In the lymph nodes, we see huge increases of T helper cells, CD4, sorry, CD8, T regulatory cells, and of B cells, both at first application and second application. And then we sampled serum and lymph nodes, and I go quickly through how this is organized to look into the proteome changes. So samples are collected, control samples, test samples, and we also built an internal standard of all the samples we have. We give them a dye, different colors, fluorescent dye, and then we mix controls and test samples. These are then separated on a gel on isoelectric point, so differences in pHs there, and then a current is applied. And in a second dimension, these proteins are separated on size. And then you get a 2D separation of your samples. This is then scanned for the fluorescent dyes being present at the gel, and in an overlay of control color against treated color, you can see then these final results. And this has been explained by the former speaker, needs then to go through more processing. You look at the peaks, you look at, you select out certain peaks, you try to identify in databases of the mass spec analysis what exactly proteins we are looking at different spots in the gels. And then you see the results here. So for the lymph nodes of the first application or second application, we have 38 proteins coming out as differential, of which we could 26 identify out of different databases. 15 were upregulated and 11 were downregulated. 58 different proteins of the 2 applications, so a little bit more. In serum, we have less hits, so it's of the first 7 and after the second 16. Just to have a notion whether or not this could lead us to some result, we did put this rough data, in fact, first in a cluster analysis, a principal component analysis, and you see the controls are here indicated in green and treated in red. For the auricular lymph nodes, we could not 100% separate out on the first principal component the 2 different groups, because one here is red still at one side and one green here. The separation is better after 2 applications, and for the serum more or less the same, although it was more cumbersome to be really find good separation of the set of data. Then, as also shown before, we looked to ontology, so the different enzymes identified, looking what is their function and to what does that lead and in what pathways does that fit. And you see here, certainly a lot is always very basic issues going on in proliferating cells, structural proteins, protein, stress responses, it's probably acute phase responses also there, both in first and second application, but also some more typical immune responses and so on. The same in serum, although here the separation is very difficult to make, and it's probably not very meaningful yet to find out what is going on in the serum at very early time point in the sensitization. And then trying to put all the data together in a pathway analysis, and I just show here a simplified pathway analysis and indicated those proteins that have been picked up from the lymph nodes, proteins that have been picked up from the differential comparison in the serum, so in green and in yellow here given, and the cytokines we did measure in the lung being present normally during asthmatic response in our mice. And then you see that we can find some links between all these different, of some of the proteins being identified. And then looking into more detail, it's a big job looking through the literature on functions of the different identified enzymes. For instance here, lymphocyte-specific protein, which is been found to be expressed in typical immunological cells. It is active as a chemotaxis activation of neutrophils, and it's regulated by, for instance, IL-4. And in a second set of mice, we also treated them. We tried to verify the different elements from the pathway analysis we found from our first set. And yes, if we look at, for instance, this protein LSP1, we see clearly not after the first application of the sensitizer, but after the second application, we see a reduction of LSP1, which is in line with the induction of IL-4 in the system. Another one, corolin 1A, which is important in T-cell activation, mediates calcium release, and here also we see that this is increased also in a confirmative experiment and a second experiment with other mice. Another one, hemopexin, is more complex, and we cannot really yet explain everything what is going on here. So it's normally involved in innate immunity, upregulation during inflammation, so an early response. And if we then look what is happening, we see that at the first application, we see an increase of hemopexin, but after the second application, we see rather a decrease in our experiments. So maybe it can be explained by this acute phase, and that it's after the second application, the acute phase is not therefore too much present, but although we doubt that is a good explanation. So to come to a conclusion, so we I think we have that sensitization due to skin contact is important, that we already showed that, and that we did confirm in our two experiments, first with the proteome analysis and then to specific endpoints as we did pick them from the pathway analysis, that what we see is probably correct, and that the proteins that have been identified as may be important in sensitization and as a marker for sensitization could be used, but unfortunately this has not been yet validated in human exposure. So therefore we should find funding and some time to do that. And these are the people which were involved, Steve and Hannah, who did the research in our group, another group which was working in genomics, proteomics, and the main person here was Elke Kleine working in our project, and the service of the university running all the proteomics techniques is led by Dr. Bagerman. So thank you for listening.